And so let's start with sort of like the major problem in terms of drug development. And it's essentially the scenario that's painted for you here would be sort of like the cost and the time it takes to take a small molecule, in this case a drug, from basic discovery to being approved for first-in-human trials. So it essentially takes, on the average, about 14 years to go from this step to that, and certainly in the order of several $billion, anywhere from $2 to $6 billion to take a small molecule, and you start with a number of different compounds, and it's really a very slow iterative process till you get to one drug that can be approved, and even when it gets approved, in a lot of cases, it actually fails. It fails in human trials, and that is because the clinical data that supported the research was kind of weak. And so that's what I'm trying to approach here in this talk today, is what can we do in this area, or this what's called the valley of death, to reduce the attrition of these compounds and essentially improve our predictive ability to know what are going to be blockbuster drugs. And so here's a study that was done from 1991 to 2000 that broke down into various components where the major bottlenecks are in terms of drug development. And as you can see here, over a period of 10 years, over a decade, the preclinical safety and efficacy and toxicity comprise the major area of concern in areas of PK and bioavailability. As you can see, in 1991 and then recently in a 2000 study, because of advances in chemistry and computational chemistry, the bioavailability issues has largely been addressed. But yet, safety and toxicity as well as efficacy remains the major problem. In fact, it still accounts for over 60% of the attrition of drugs. And the situation has actually not improved much. So most recently, a study done in 2011 showed that the success rate has not really improved much. So efficacy still remains a problem, as well as safety, still accounting for, at this point, 65 to 70%. And you can see where the various failure is happening both in cancer, in neurology, cardiovascular diseases, GI, and other systems. So in a number of instances, the scenario has not really changed through the decades or so. And so what we have identified at the NIH is really that the problems can be broken down into three major parts. And that's what we call things that are being lost in translation. So there are many published preclinical results that are not reproducible. And so what we need are more robust and repeatable assays. And then also the animal models that are being used to predict human response is also not being predictive. And that is not surprise. Humans are not mouse, or mouse are not humans. And so physiology, human physiology is different from mouse physiology. In fact, there was a study recently done on sepsis that showed that 20 years of research on sepsis using animal models, mouse models for sepsis, have not produced any drug at all, resulting in certainly loss of time and billions of dollars. And then the last area that we have identified is that the FDA, while they are the gatekeepers for approving drugs going into the clinic, they're still using 30-year-old biochemical approaches in terms of assessing safety and toxicity. And so we are working with the FDA in terms of updating their assay systems, and that is what I'm going to be describing to you about our tools and technologies that will address, hopefully, these three areas. And so what we essentially need to come up with better approaches for drug development would be better tools that are highly predictive of safety and toxicity, better tools that can also show us efficacy, which right population to treat, so essentially stratifying the population groups. And then efficiency, being able to reduce a lot of the false negatives and even the false positives that might come into the clinics. And so what we need are really highly predictive tools. And that is where we need to be approaching these issues. Now, the issues I've described for you in the last few slides are essentially for a lot of diseases, but the needs for rare diseases are not too different. You know, in terms of rare diseases, and certainly they're rare because they affect 6 to 8% of the world's population. There's about currently 7,000 rare diseases that have been identified, and only 5%, less than 5% of them have effective drug therapies, and most of them are going to be in the order of stem cells, some protein replacement approaches, and stem cells. But yet, there's also a broad spectrum of illness and etiology, including genetic rare cancer, congenital malformations, autoimmune responses, and toxic and infectious and degenerative processes that can all lead to what we consider rare diseases. What constitutes as rare? It's about 600 to 700 million people worldwide are known to be affected with rare diseases. And then depending on which region, geographic region you're at, even the definition of rare can be different. So in the United States, it's less any condition that's less than 200,000 as per the Orphan Drug Act. In the EU, I believe it's one for 2,000. Japan is less than 50,000. And you can see that some of these are incidents and some of those are prevalent rates. And so it's, even for different countries, it's different. And so that makes the approach of treating rare diseases even different. But yet the challenges for rare disease research are also quite unique in the sense that, of course, you have a small number of patients. They're typically widely dispersed geographically and often quite familial. They often lack a patient registry system, so it's hard to recruit. Families, lack of information, availability for families and providers, different definitions, regulations from region to region. There's also lack of animal models, and because we have no animal models, we can't study essentially the pathophysiology of the disease. And so because of the above areas being geographically dispersed and small number of patients, quite often we don't have a good understanding of their natural history or the progression of the disease. And so there's lack of historical data. There's also variable age of onset, no biomarkers available, and essentially clinical endpoints. Because of the lack of natural history, we don't have necessarily a good sense of what the clinical endpoints are going to be, what we're aiming for in terms of treatment, uncertain durations of therapy, heterogeneity in treatment effects, and absence of placebo controls, because again, you're working with a limited number of individuals. But yet, the issues in terms of therapy development is not necessarily different within a common disease or a rare disease, in a sense that whether it be a non-orphan or a rare disease, you still have to determine the effectiveness of a drug to the patient, whether in treatment or in prophylaxis. You have to assess the risk and safety benefit to the individual. Determine the overall risk-benefit to patients who will be treated with the drug for a given condition, and should certainly have to have data that's capable of meeting the regulatory requirements to advance that drug. So that's just because it's rare doesn't necessarily mean that they're exempt from any, although there are efforts within the FDA to fast-track some of this approval process. And so here's where the National Center for Advancing Translational Sciences came about. This is the newest organization within the NIH. It's only about 2 years old. So I moved from neurology to this new center, having been recruited to lead the special office of, the Office of Special Initiatives, and essentially the office of, the National Center for Advancing Translational Sciences, or NCATS. Our mission is not working on any specific disease. What we're looking for are identifying issues or bottlenecks that underlie a number of disorders, and identifying the common problems, the common bottlenecks, and helping address that so that we can have a larger impact. So what we our central mission is to generate innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. Because as I have said, whether it be a rare disease or a common disease, problems are, the common problems underlie those issues. And so what we're seeking to address here are where the problems are, providing tools and technologies that will address those problems and helpfully address rare and common conditions as well. The program that I'm describing to you is not necessarily new. The NCATS has a number of different programs within the NIH that addresses rare disease. There's rare disease research. So we actually have an office dedicated to the study of rare diseases, and they have a number of programs that you may have heard of, the Bench to Bedside Awards, the Rare Disease Clinical Research Network, or RDCRN. We have a rare disease human biospecimens biorepositories, the Undiagnosed Diseases Program, which is largely a sequence-based approach to diagnosing diseases, setting up a global rare disease patient registry, and then as well as a data repository. And then there's also programs called the Therapeutics for Rare and Neglected Diseases, and then Bridging Interventional Development Gaps. These two programs essentially are looking at advancing, once you identified your small molecule or your therapeutic, it's really helping the investigator get to the preclinical and safety and toxicology data that you would need to present to a regulatory agency to get first-in-human approval. And then this is the new therapeutics use. It's essentially, we call it drug repurposing, taking already approved drugs and seeing if the condition will work or the approach will work for other indications. But what I'm going to be talking to you about today is a different program than the ones I've talked about. And this is called the Microphysiological Systems Program, or MPS, or otherwise known as Organs on Chips. And so our challenge here is to develop essentially what we call biomimetics. So these are representative of human organ systems, and they are to be represented with human cell types. So we have a 3D cellular architecture and should represent in vitro physiological function in this organ mimetics. And so let's say for the liver, if we have a liver on a chip, that liver on a chip should metabolize drugs and vaccines just like a liver would, and as well as physiologically synthesize albumin and bile. Let's say if we're developing a lung on a chip, then it should permit the gas exchange between air and blood. So there should be an air-liquid interface, be able to synthesize surfactants, as well as some of the different markers for lungs. And so that is, in some ways, a challenge. And this is something that we launched 2 years ago in terms of how to go about doing this. And this is really where convergent science comes about. So this really takes really a multidisciplinary approach where we're taking in different disciplines, different tools and technologies from different fields of expertise and bringing them together to bear this into fruition. So in some ways, we're calling this common building blocks. So we start with what is the best in terms of what we know about the extracellular matrix, what are the best in terms of material science that would be surfaces that will be biocompatible, that will allow the growth of cells. And then, of course, we start with our source of cells. And in this case, we're very much interested in using stem cells and progenitor cells that will be able to be differentiated into multiple tissue types. And that is important because we want this to be a renewable resource. We also want this to have known genotypic composition, so we can represent individuals from different population groups and have that represented essentially in terms of the different organ systems. So we don't have incompatibilities in terms of, let's say, a liver on a chip from one individual versus a lung on a chip with another individual, because our hope is that these are not going to be standalone devices, that we're actually going to be linking all these organs on a chip to form a human on a chip system. And I'll be talking about that later on. And we also need some idea about the porosity and the topography and stiffness, and this is where a lot of the biomechanics fall into place, because like I've said, we need to represent this into a 3D architecture, so we need to have the right surface area to allow growth in a three-dimensional way to support the different cell types, and then the right gradient, both in terms of cytokines and some of the growth factors that mimics both spatial and temporal patterning. And then, of course, when you're developing a 3D culture system, the issue of perfusion comes about. You know, how do you mimic the flow of nutrients and as well as the outflow of those toxic end products? And we can essentially achieve that either by microengineering or by microfabrication through microfluidic channels, or actually inducing stem cells to actually produce vascularization into these different tissue types. And we have projects that are doing both. And then the next area that we need to integrate would be some idea of what it takes to optimize the culture conditions, what are some of the biomechanical properties, the kind of stretching, strain that are needed, and essentially a blood mimetic that is needed to support different cell types within a culture system. Because as you may know from some of the culture systems, some of the cells can be fastidious in terms of how they grow, and so they can be very specific in terms of the growth media that they would need. But if you are now combining multiple cell types to represent an organ, and then on top of that linking different organ systems, then the challenge grows in magnitude. And then we also want to show innervation into this microphysiological systems. And then not only innervation, but immune response as well. And then essentially to have a real-time, label-free, non-destructive way of assessing what's going on in this bioreactor system. So because these are, again, going to be very small devices, and I'll give you in a video a short flavor for how this looks like. And so this is very small, and so we can't afford to lose cells or tissues. So we want to be label-free as much as possible. And then lastly, to have built into the system a way, iterative way, so that we can continually improve on the system through some of the computational designs and integration systems. And by the way, feel free to interrupt me if you have any questions. And so this is the program that we came up with 2 years ago. It's called the Microphysiological Systems. Again, it's to develop an in vitro platform or a biomimetic platform that uses human tissues to evaluate efficacy, safety, and toxicity of promising therapies. And our goal is to have the 10 major organ systems represented. So that would be the circulatory, the endocrine, the GI, immune, the skin, musculoskeletal system, the nervous system, reproductive, respiratory, and urinary system. We want this to be physiologically relevant, genetically diverse, and pathologically meaningful. We want this also to be modular and configurable. Like I said, these are right now being developed as individual organ systems, that we want to fit them together kind of like Lego blocks, to be able to configure one and line them up based on the type of analysis that one needs to do. And then also the ability to keep the cells alive for at least 4 weeks of testing. And then, of course, at the end of the 5 years of the program, to make it available to the larger research community. As I said, this started 2 years ago, so that we're actually 2 years out into the program. And so I'll be giving you some updates in terms of where we're at into the program. It takes a large cooperative efforts within the NIH, primarily through NCATS, to realize this program. And so the NIH, through NCATS, has invested about $75 million over 5 years for this program. We also were fortunate to be able to partner with DARPA from the Department of Defense. So I don't know if you're familiar with it. It's the Defense Advanced Research Program Agency. It's essentially the go-to agency within the Department of Defense to come up with some of the innovative tools in engineering. So they came out with, for example, with the GPS, the neural prosthesis arm, some of the robotics that comes about essentially to power up what they call the warrior system within the United States military service. And then, of course, we have a partnership with the FDA as a regulatory advice, but essentially their role is a non-funding agency, but essentially to provide us with regulatory advice and toxicology expertise. And so the way the partnership works is we share expertise and materials amongst all the investigators. We hold joint semi-annual meetings. We provide a common set of validation compounds, and we facilitate collaborations. So essentially what we have is the human organs on chip from the Harvard Institute, and then it's called the biomedics from the Massachusetts Institute of Technology. And then we have funding that was made to a number of different academic institutions all over the United States to come up with organs on chips for the lung, for bone, for muscle, for skin, the brain, the blood-brain barrier, integrated blood-brain barrier plus the GI system, the gut, the female reproductive system, kidney, liver, heart, liver, and vascular system, heart, lung, and all sorts of different combinations. And then we also partner with biotech companies like Nortis, Kayatech, and CN Bioinnovation to make this essentially tools commercially available. So this is sort of like a depiction of what the system would look like. This is a lung on a chip that was developed at Harvard, the Wyss Institute. So that essentially is the lung on a chip. As you can see, there's fingers holding the chip itself, so it's no bigger than a USB stick that you can have. And essentially the real estate platform is here. And what we're trying to represent is the breathing lung, where you can see that in this case, the functional unit of the lung is, of course, the alveoli, which undergoes a lot of stretching. And of course, there's air and liquid interface, as I'm showing here. Here's a cross-section of what the lung on a chip looks like. Essentially, it would have a microfabricated membrane system. It's a PDMS membrane system that's porous. It's got pentagon holes drilled into it. And then we culture lung epithelial cells on the airway side and endothelial cells on the capillary side. And then on each side are vacuum channels where you provide cyclic vacuum. And so it promotes the stretching of the cells, so like simulating the breathing system. And then you can flow air on the airway side and blood or nutrients on the fluid side of things. Now, in order to show that this is actually physiologically relevant, you can flow bacteria into the airway side, and then white blood cells on the endothelial side. And just like what your normal cells would do when they sense an infection, they would actually go through diapedece's and engulf the bacteria. And this, of course, this is a video representation, but this is a real-time video where the white blood cells are labeled and they're shown flowing. But once you start flowing bacteria on the airway side, then all of a sudden they will start sticking to the endothelial side, as shown here. And then you'll see a high-resolution image of a particular white blood cell that is making its way through one of the pores, and then making its way through the pore, and then engulfing the bacteria on the other side of the system. So this is just to show how it works also in terms of drug development. Again, using the same lung on a chip system. One of the things that is lacking in terms of a model system is edema. And so pharmaceutical companies are very much interested on working with models of edema. but there are no animal models, nor certainly in vitro models for edema. But one of the common side effects too of IL-2 chemotherapy, which is a common chemo drug for kidney and melanoma, is edema. And so in this particular case, if you do a FITC labeled inulin and flow it on, as shown here by the green particles, flow it on the endothelial side, as well as with IL-2, and then we measure the migration of the FITC labeled inulin into the airway side. That will show us to what degree we're modeling edema. So in this particular chart, it shows that if you have just the IL-2 by itself with 10% stretching or strain, it actually show leakiness, shown here by the fluorescent labeled inulin going into the airway side. But if you then introduce a candidate drug, but known by its properties to at least hopefully mitigate the effect of IL-2, as shown here, it prevents the formation of this edema. And so the drug manufacturer, in this case Johnson & Johnson, is very much interested in making this the drug A as a companion drug to IL-2 chemotherapy. And so that is one of the examples in terms of how this can be used, this organs on a chip or lung on a chip or alveoli on a chip can be used to represent a specific human physiological response. Another device that's also from the Wyss Institute, this is now from Kit Parker's group at Harvard, is to show that you can also mimic the contractility of cardiomyocytes on these devices. So you can essentially etch out, culture iPS-derived cardiomyocytes, and then etch it out so that it forms this cantilever device, and then it actually undergoes spontaneous beating. And so you can see here that it's actually very, very rhythmic in terms of its contractility. So you can measure the contractility of this and then be able to show drugs that will have either induce cardiomyopathy, or in an example that will show for rare disease, show that certain conditions can actually be simulated in this heart on a chip system. We also have projects that we're funding in terms of neurodevelopment. So this is from Jamie Thompson's group at University of Wisconsin, and his project is to come up with an in vitro brain on a chip system. And so what he has here are essentially iPS-derived neural cells that he was able to differentiate into astrocytes, GABAergic, glutamatergic neurons, as well as endothelial cells and pericytes. And he was able to co-culture this and be able to form, and they actually self-aggregate to form this nice formation of, including the vasculature, glial cells, and nuclei, and actually using transcriptomics show that there is actually synaptic formation and communication within all the different cell types. Then we also have some funding to come up with a kidney on a chip, and this is from Jonathan Hillman-Farb's group at University of Washington. In this particular case, also looking at primary human epithelial cells, and he was showing that you can actually take known kidney markers to show like ZO1, E-cadherin, aquaporin, kidney injury marker 1, KIM1, and show that it actually is represented in this kidney on a chip system. And so what the project is to come up with the proximal tubule of the kidney, because that is the site where most of the renal toxicity comes from, and be able to actually have brought with me a kidney on a chip. So anyone's interested, I have a small device that I can show you after the seminar if you want to see how it looks like. But essentially what we have for the kidney on a chip is a representation of the proximal tubule of the kidney. And then we're also funding Lance Taylor at the University of Pittsburgh to come up with the liver on a chip. And of course, as you know, the liver is not just a group of hepatocytes, but actually it's a very complex group of cells in terms of having stellate cells, Kupffer cells, along with the hepatocytes, and then being able to have endothelial cells through the fenestra represented here. So that's the challenge in terms of representing that. And so he has that also represented on a chip system. But what is also important with the liver on a chip is the incorporation of biosensors or sentinel cells. These are essentially optogenetically labeled cells that will allow us to detect in a non-invasive way how the cells are behaving or how they're physiologically reacting to a particular challenge, whether it be biochemical or metabolic. And this is just a way of reading it, and it's done through a high-content imaging system where the fluorescent detection is being used to determine if the biosensor cells are giving us the correct readout. And so in this particular instance, this is an example of a biosensor detecting the release of cytochrome C, an indicator of apoptosis when the mitochondrial integrity, membrane integrity is being detected. So in this case, that's for cytochrome C release. There are also biosensors for nuclear and cell positioning, for reactive oxygen species, for mitochondrial calcium uptake, steatosis, biocanicular efflux, and oxidative stress. So these are all biosensor cells that have been labeled with different fluorochromes and essentially co-cultured with the liver on a chip and allows us to have a non-invasive way of determining the status of the cells based on particular challenges. We are also making available on this program a group of training and validation compounds. So we have collected about 100 plus compounds with known toxic and biologic responses. And so this essentially are compounds that have known to be safe in animal models or in vitro testing, but when put in humans have shown to have various adverse events or side effects, and so have allowed the withdrawal of the drug. So we collected those samples. We're providing it to our investigators and challenging them with the task of, can you predict better than an animal model that these compounds will actually show the toxicity that was not predicted in animal model system or in vitro system. And so that will be the challenge. And then we're also providing them with validation compounds, which the investigators are going to be blinded to and will not be, and of course we know what the outcome should be. And so the challenge then is to demonstrate to us that the microphysiological systems are actually behaving in a physiological way. We're also having a contract with Coriell Institute to come up with some of the quality standards for IPS. Anyone who's working with IPS or stem cell technology knows that this is a big deficiency in the field right now in terms of what are the quality matrix by which to evaluate stem cells. And so we have come up with standardized characterization criteria, including sterility, normal growth conditions, karyotyping, embryonic body formation, and then I left out a number of different things where you're also doing whole genome sequencing analysis on this particular stem cell types, stem cell lines, and these are the same stem cell lines we're providing to our investigators to start out as starting materials. At the end of year 2, as I mentioned, this is now year 2, so we have standalone devices for each of the organ systems that I've identified. And the project has so far shown that the organs and chips has shown functional and physiological relevance. They've been tested with a training set of compounds with known toxicities, shown viability from anywhere from 3 to 6 weeks. Then we have non-invasive, non-destructive readouts, and essentially have been incorporating human-induced pluripotent stem cells and progenitor cells into the platforms. And then there's also QC and characterization of the stem cells, training set of compounds. And this field has also produced mini pumps, microanalyzers, database biosensors that we're making available to the general community right away. And so because again, this are essentially coming up with new technologies depending on our needs, and then also improve biocompatible scaffolds to support the 3D organ systems. So these are just representations of what those organ systems look like. So eventually, at the end of the day, at the end of the 5 years, what we want to do is be able to, like I said, plug all the different standalone organ systems into sort of like a viable system where you, let's say, if the drug you're testing is an orally bioavailable drug, you connect the gut on a chip with the liver on a chip, and so the drug gets metabolized, because some of the toxicities can come out from liver end products, and then it goes to the kidney, to the bone, to the heart, and to the lung, and so you're able to then look at individual organ toxicities or even multi-organ toxicities. You can actually even redirect some of the flow to find out where the toxicity is coming from, whether it's organ-to-organ interaction or a drug-to-organ interaction. And that's what I mean by a plug-and-play system. And then eventually what we will have would be a human body on a chip that we're able to develop all these readouts and essentially make the in vivo correlation in terms of drug toxicity and metabolism. In terms of application to rare disease, this is one area that I'll show you. This is an application to Barth syndrome. It's an X-linked recessive disorder caused by mutation in the tafazin gene located in XQ28. It's characterized by severe cardiolipin deficiency in the mitochondrial membrane and characterized by cardiomyopathy, neutropenia, skeletal myopathy, growth deficiency, and 3-methyl-glutaconic aciduria. It's unknown in terms of what the actual incidence rate is, but it's estimated to be one case out of approximately 1 million births. And essentially, the project this was just published about 2 weeks ago from Kit Parker's group. Essentially, they took iPS-derived cells from patients. And so these are patients with a frameshift mutation or a missense mutation. And then what's important too, also as a control, is you took fibroblasts without the mutation, derive iPS cells from them, differentiate them into cardiomyocytes, and then from the patient, you can also introduce mutations so that you can work with an isogenic background and introduce the same frameshift mutation and the same missense mutation in that, and be able to show that you're developing the same deficiencies, cardiomyopathy deficiencies. And what's more important is that the paper also showed, this was published actually in Nature Medicine. This is an editorial that came out in Stem Cell on the topic, that you can also evaluate potential therapies like linoleic acid, which is a precursor to cardiolipin, or mitotempo, which is a radical oxygen species scavenger, and be able to show mechanistically what is happening in these patients. So this is some of the data that was published. So this is a profile of cardiolipin. So this is the precursor to cardiolipin in control, and what the cardiolipin looks like as a profile. So you can see that in patients that there is a cardiolipin deficiency in the patients. And then you can look at this quantitatively also in terms of the wild type, and then the two mutations, both the missense and the frameshift, showing that there's an elevated level of precursor to cardiolipin ratio in these patients. In terms of physiological activity, if you do alpha-actinin labeling, this is how the cardiomyocytes look like. They essentially line up isogenically, but when you look at the beating as measured by the thin film that I showed you, where it's measuring contractility. It shows you that the bars in dark red are the ones that are derived from patients with the missense mutations, and the ones in light pink are the ones that have been corrected through gene transfer, and shows that there's at least a restoration of that contractility. And then this is how it looks like histologically. This is from a wild type. This is from a patient where the sarcomeres don't line up very well. And this is from a patient line that's been corrected by gene transfer, and shows that there's correction also. And this is, again, from a patient line. So at least from histological and physiological standpoint, you can represent Barth syndrome on a chip in this particular case. And this is a slide that shows different therapies, candidate therapies. Again, this is with the gene correction. This is with linoleic acid, approximating the gene correction level. This is with another form of lipid, and then this is arginine and cytosine, showing also partial correction. And so it allows one to evaluate the some of the potential therapies in the system. So what I have shown you in this organ on chips, this giving you an overview, is that this is a 5-year program. Again, with NIH, DARPA, and FDA, we have the goal in 5 years of achieving a 10-organ system mimetic that is physiologically relevant, similar to a human body on a chip. As I've shown, can be used for preclinical safety and toxicity assessments. And essentially for human disease, we can also model human disease such as Barth syndrome. We're also in the process right now working with patient groups in terms of modeling cystic fibrosis, for example, on the lung on a chip, and then also progeria on at least the aging of the vascular system on a chip, showing the stiffening of the vasculature for progeria patients. So we can essentially model human diseases in this. And our hope is that this will provide a sufficient alternative for animal testing, and that eventually we're going to build up a diverse group of IPS lines, and essentially that can be used for clinical trial settings. And so this is actually a lead-in into that. So if you have, let's say, donor cells, fibroblast cells from thousands of normal individuals. So there's actually George Church from the Harvard Institute of Genomics has already 16,000 fibroblast lines that he's collected from various individuals that he's doing whole genome sequencing on, and he is collaborating with the New York Stem Cell Foundation to turn those into IPS lines. So you can have potentially upwards of 15 to 20,000 individuals fully sequenced on IPS lines that can then be differentiated into various tissue types or cell types, whether it be cardiomyocytes, and you can essentially take individuals with diseases and introduce mutations in an isogenic background. You can do particular individuals, let's say with a rare condition, and make your own, Dr. Camella's lung human on a chip system, and use that as a personalized medicine approach to understanding what best drugs to test for your own personalized human on a chip system. And we can use this for drug optimization studies, as I mentioned, for disease modeling. And essentially where DARPA is interested in is essentially to test countermeasure agents. I didn't show it here, but we can actually culture the microbiome or a gut on a chip system. So even up to now, most individuals can have difficulty in culturing one type of microbiome in a system, whereas in our gut on a chip system, we can actually culture pretty much entire flora on the gut on a chip, and actually have shown that it's essential to forming microvilli and crypts on the gut on a chip system. And then we can then test for a number of toxicities. As I've mentioned, if you have tens of thousands of individuals with different genetic compositions, one can then conduct at least early phase clinical trials on a chip in the future and be able to identify candidate therapeutics in a better way. This is a large group of individuals that are involved in the management of this group. From DARPA, program manager there is Barry Pallotta. From FDA, we have a number of different advisors providing us input in terms of toxicity testing that's needed for regulatory approval. And then from NIH, provide the oversight and leadership on this program. And then we have representations from various institutes and centers at the NIH with specialties in particular organ systems, because this is truly a cooperative effort. And then this is where we hope that this will lead to. This is actually from a figure that we published in Experimental Medicine that gave an overview of the paper. So essentially what we have is future applications in terms of study of the microbiome, toxins, infectious diseases, as I've said, countermeasures, and then tools for clinical trials, including conducting clinical trials itself on these chips. And so with that, I can end and take questions if there's time.